So this video is about ligand-gated ion channels or ionotropic receptors. So here we will learn some of the key features of ligand-gated ion channels and their mode of action with the help of examples. Ligand-gated ion channels are sometimes called as ionotropic receptors. Some of the key features of ligand-gated ion channels are as follows. Firstly, ligand binding regulates the opening of ion channels, allowing the inflow of positive or negative ions. Inflow of positive ions induces depolarization and inflow of negative ions induces hyperpolarization, resulting in a cellular effect. So how do we differentiate ligand-gated ion channels from voltage-gated channels? So in voltage-gated channels, the opening of the channel is regulated by changes in voltage gradient across the membrane. Examples of voltage-gated channels are sodium, potassium, calcium and so on. Whereas ligand-gated ion channel is a receptor which operates through the opening of channel in response to binding of a chemical mediator. Secondly, these receptors are directly linked to ion channels and do not use second messengers, unlike other types of receptors which involves signaling pathways to produce cellular effects. Thirdly, these are the receptors on which fast neurotransmitters act. They are involved mainly in fast synaptic transmission. Examples of these receptors include nicotinic acetylcholine receptor, GABA receptor and glutamate receptor. Fourth, ligand binding and channel opening occurs on a millisecond time scale. Nicotinic acetylcholine receptor is a typical ligand-gated ion channel that plays an important role in transmission at neuromuscular junction. It consists of 5 receptor subunits, that is 2 alpha, beta, gamma, delta subunits with transmembrane helices arranged around a central aqueous channel. There are 2 acetylcholine binding sites in the extracellular portion of the receptor at the interface between the alpha and the adjoining subunits. When acetylcholine binds, the kinked alpha helices either straighten out or swing out of the way, thus opening the channel pore. This results in a net inward current carried mainly by the sodium, which depolarizes the cell and increases the probability of generating an action potential. The next example is GABA-A receptor. Its endogenous ligand is GABA, which is a major inhibiting neurotransmitter in the central nervous system. Binding of GABA to the receptor causes activation of receptor, causing opening of GABA-gated chloride channels. Inflow of chloride ions causes an inhibitory effect on neurotransmission by diminishing the chances of generation of an action potential. The next example is glutamate receptor. AMPA, NMDA and kainate are glutamate ionotropic receptors. Glutamate binding causes activation of AMPA receptor, causing opening of sodium channels, allowing inflow of positive ions and causing depolarization of the cell. Depolarization of the cell sends a signal to NMDA receptor which unblocks the calcium channels, allowing the entry of calcium ions, which is responsible for slow excitatory response. So AMPA and kainate receptors are involved in fast excitatory neurotransmission, whereas NMDA receptors mediate slow excitatory response. These are my references. Thanks for watching. Please like, subscribe and share.